Hi, this is Dr. Jabali, and in this video we're going to focus on topic 2.6, which is all about DNA and RNA structure. We're going to go through some previous IB exam questions, so let's go forward. So here we have always look into the command word, which it says describe. So this, whenever you see the word describe, you can write in words, but you can also do drawings and annotations because you can get points from those. And here we're talking about the structure of DNA molecule. So here we can do the drawing as well as writing, but I'm the type that sometimes I like to prefer to drawing because that's much easier, more visual, but some people want to do the whole description writing, and you can do both. So for me here, when I have to do a DNA molecule, a lot of students will start thinking that they just have to draw this, and then they have to put A and the T, and then the C and the G here like this. If you do this kind of drawing, you are not going to get any points. Neither are you if you just draw one nucleotide on one side and the other nucleotide on the other side. For example, right here with the drawing going on of a phosphate. This is not enough. You need to put more information, more in detail. So usually I'll tell students to at least make sure two nucleotides on each side and make sure you label everything. So how would I make sure that I am getting all the points? The first and most of all is make sure that you draw the nucleotides really well. So here I will have the phosphate bound to a sugar, which is the sugar usually looks like a house. And you can see that it's very important that the phosphate is bounding on the top of the house so as the nitrogen base is right on the tip of it, and you have it right here. And then I will connect through the base again to another phosphate, and then a phosphate to another sugar. And then here on the corner. Now it's very important that you can see where the location is actually on the tips of, if you think of the roof of the house, this is where the nitrogen and the phosphate is bound. In this case, I would always label the things. I would put phosphate group, and then I will have here written nitrogen base. And of course, since it's the basically sugar or which is DNA, I will not write sugar here because that's not usually going to get a points. I would actually write the deoxyribose. So I got the points on this one side, but it's not enough. I will put letters here. I always make sure I put both types of letters. See here. Both types of letters. Now I have to do the opposite, so I will start with the nitrogen base to keep it more neat, and I'll make sure I put T. The next thing I would do is make sure I put the hydrogen bond, and it has to be dotted line. So there's two of them, and this will be known as the hydrogen bond. Now just right, you also have to label it, so make sure you label and touching the line right there. You also have to do 3 dotted lines because it's three bonds when it comes to C and G. And again, it's the same thing. It's a hydrogen bond. So you can just line and touch the line here and say hydrogen bond. So now we have labeled everything from phosphates, the sugar, the nitrogen bases, everything. But this is not enough. You have to complete the other side of it. So the other side, if you notice, the sugar house has to be going upside down now. So we put it upside down, and we can put it here also, the upside down. And then we have to connect the phosphates again to the two sugars, and we can add a phosphate here and a phosphate here. Now we don't have to label on this side because you're already labeling the other side. Now what I can do with this also, if you're an HL, is put the letters, right? So I can put the letters here. So basically one side is going to be 3' to 5'. So we know that this is carbon 1, this is 2, this is 3, this is 4, and there's a 5 here. So this is a 5', and this is a 3'. Well, this is a 3' right here, and this is a 5'. And you can say that DNA is a double helix. And you can also say that DNA is anti-parallel. So basically, you're describing everything you can think of about DNA. Drawing the structures, the labels, the complementary base pairs, which you can also do this. You can highlight these two together and say they are complementary base pairs. You're also going to get you're going to get answers for that. So if you notice here, I described in the drawing, I didn't have to write, but it's very clear that I am showing everything you can think of. Like I did mention that it's double helix. I did mention it's anti-parallel by showing the numbers right here. I have labeled the sugar, the nitrogen base, the phosphate group. I have shown the hydrogen bonds, triple and double for AT and CEG. Now, if I want to go more in details, I can always write the words for the letters A, T, and G, C, which in this case would be adenine. And then we will have thymine. And then we have cytosine. And then we have guanine. So you write, this right here is everything you can possibly write when it comes to drawing the DNA molecule. So you can take a screenshot, make sure you're practicing and writing this, and you get a full point for sure. So let's look at the mark scheme very quickly here. See, it's a 2-strand double helix, and if you see anything that is underlined, like in this case here, that means it must be set. And I did mention here, so which that means I definitely get the points. Otherwise, you're not access. So if you just say double, if you say double without the helix, it's not going to work. I did mention it's antiparallel, or I can show it the strands, right, which I did. I did both ways. I show the sugar phosphate backbone. Each strand is formed by chains of nucleotides. Now, I did not mention this, which I haven't mentioned here, but I could actually do a highlight like this, and I could mention that this is a nucleotide. Or I can just make a written sentence that says DNA is made of nucleotides. And I mentioned phosphate, deoxyribose, and a base, and it can be annotated. As I mentioned, I did draw it. The base is adenine, guanine, cytosine, thymine, I have mentioned. Strand handled by the hydrogen bond, I have mentioned, and I did pair the A with T and the C with G. Now here is things that you need to know. Look, both helix and 2 strands need for AMP. So I did show the 2 strands and the helix. Here, although it says the double helix is sufficient for a mark. Now the points can be awarded for annotated diagram, which I did. I'll draw. For MPC, which is the sugar backbone, a label of sugar phosphate is required. Now to award the MPD, which is this, from A diagram, at least 3 pairs of nucleotides must be shown, which we did show 3 pairs of nucleotides. And then from MP, a diagram would need to be labeled as nucleotide. So I did it right now. Expect the deoxyribose, not sugar. As I told you, they're not going to accept the sugar, so you have to mention the word deoxyribose. Write the names of the bases required for the MPF. So for this, you have to mention all of them. You have to write the full words for these 4 things. Don't just put A, T, C, and G. This is like when they're being, IB is very picky. So I always tell students, really write the whole words. Don't just write A and T. Do not penalize twice for MPF and G using letters. So these 2 right here. So that means when I'm writing those words and then I'm putting them on the chart, which I did, right? I wrote the words here and I put them in the chart. I'm not going to get penalized for rewriting them again. If only they use the symbols. So here he says, if they're only ever using the symbols HCG, they are ineligible to APF. So this means right here, let me highlight this one. I don't get points unless I write the full names. That's why I tell you, write the full names. However, if A pairs with T and C pairs, they will get. So that means I did mention here there are bonds going on between them, so I can get the points. So I'm always playing the safe side. I will write the entire names in there. I will present the nucleotide as it is here. I will write the deoxyribose, the nitrogen base, everything written names. I will also say double helix, antiparallel. I even make sure to just say the complementary base pairs and I put the numbers. So this is a full scoop of writing a drawing of a DNA. So be very picky when you're drawing DNA. Do not be very generalized. Here's another question that is also saying to draw the structure of the DNA showing the possible bases. It's just like this one, but I want to show you the mark scheme, how it's very close. Here we have a sugar, a phosphate base linked correctly to form at least 1 nucleotide, which we did. For the MPA, ignore labeling the subunits of the nucleotides. Carbon atoms in the deoxyribose do not have to be numbered, but the phosphate group should be linked to C5 and the base C1, which we did. That's this drawing right here. I have mentioned the phosphate on the C5, and I have put the phosphate close to the C3. You can see here the phosphate is on C3, and it's located on the C5 here. Now look at this. Shapes other than circle and rectangle should be used for phosphate and base, which it can be easily done. Now, here is underline. Underline must be, that means it must be on your mark scheme. Deoxyribose, phosphates, they must be labeled at least once. Adenine, see, you have to write the entire word. Adenine, thymine, cytosine, guanine, they all have to be written words. So they have to be full names in order to get the points. okay? They don't care if the size of the purine pyridines, but you have to write the full names of A, T, C, and G. Two apparel chains and nucleotides linked by hydrogen bonds. That's actually drawn here. You can see that they're going in the opposite direction, and in the middle you can see so you can see here they're going opposite direction, right? They're going from a 5 to a 3, and here 3 to a 5, and you can see I did label the hydrogen and showing their base pairs. And then of course, continue here, we have all sugar bonds in correct position. Bonds should be connecting C3 deoxyribose to one nucleotide of phosphate, which that's what I was talking about here. If you notice, my C3 here is connecting to a phosphate and my C5 is connecting to phosphate, while the C1 is connecting to a nitrogen base. So it has to be very, very clear. So 2 nucleotides in each strand is sufficient. So this is another type of drawing. So both of them could be answered by this drawing that I have here, which has everything. So if you draw this as many times, like 4 or 5 times, you actually can get them all straight up into your answer. Now here in this question it says draw a label diagram, and it's showing now the RNA nucleotide and how they are linked together to form a molecule of RNA. Now RNA nucleotide is only one strand, so this means I am only drawing, in this case, one side of it because it's only made of one strand. So I can continue this in the pictures, and I'm doing the same thing, but here now I'm writing ribose, and this is a nitrogen base. In this case, I will put U because I'm trying to prove to them that there is U instead of T. I can also put the phosphate here. I can also put the O here because that's what makes the ribose different than the oxyribose. Now, how are they linked together to form a molecule of RNA? This, I could draw the DNA here, and I can put A, T, C, G, A, and then the mRNA, I would say this is U, A, G, C, U. This is proving the connections that I made from that. So I have explained. Let's see what they ask. So he says here ribose is drawn as a pentagon and labeled a sugar ribose. I did that. The base is drawn correct, linked C1 of ribose and labeled to the nitrogen base, which I did that. What I mean by C1, we're talking about C1 here is connecting to the U. Phosphate, I drawn correct link with C5 of ribose and labeled phosphate, which I have it here, labeled phosphate. 2 or more nucleotides drawn with correct link C to 5, which I did. We have here connecting between them, and the same thing here. So I have drawn the entire RNA structure. Now, this question here says distinguish between the DNA structure of DNA and RNA. So distinguish means let's talk about the difference. And on this one states 2 structural features that differ RNA. Now, if you notice, both of them are asking for structure. So I can always make a table of DNA and here RNA. And what I can see here is DNA is made of 2 strands. That's a structure, while this is when you have 1 strand. Another structure is that this has A, T, C, and G, and this is U, A, U, C, and G. So you can underline here to show that you know the difference. You can also say that this is a double helix, if you want. That goes with the 2 strands. Now, beside all this, you can say that this has deoxyribose, right? That's the sugar. And this one is just ribose. So you kind of already have like 3 examples, 1, 2, and 3. These are 3 examples showing the differences between them because that's what it's asking only for. So let's look at the mark scheme. So here is double-stranded. So since they have 2 questions, right, we have the 2 mark schemes. For the first one, double-stranded while single-stranded. Then it contains deoxyribose while it's going to ribose. This has thymine found instead of uracil. And you can say one form of double helix, and there's different forms. So this is a 4th one if you want to add these different forms. Now look at this one. This one has a number of strands, which we said 1 for RNA and 2 for DNA. Base composition, which uracil is for DNA and thymine for DNA. And pentose, which is ribose for this, and deoxyribose. So you can see that the most common ones are these guys. We're talking about the strands, the uracil for the thymine, and the sugars. That's exactly what you have here for the strands, the sugar, and the uracil. So that's the most common things that you have to put in mark scheme when comparing DNA and RNA, and you're certainly going to get the points for it. Now, this question explain the model. The model we're talking about, Watson and Crick. Now, Watson and Crick, they actually, what they did was steal an X-ray from Franklin Roosevelt and Wilkins. And what they did was take that design, and what they did was look at that design and try to create what would look the shape of DNA. So they took the information of an X-ray that was created, right? This X-ray. So you must mention the word X-ray diffraction, because that was what results came out from it. And then from this X-ray, what it did was build a model, right, of the DNA. That's important that you mention the model, even though it's here, you mentioned the model of DNA. And they tried to fit the model DNA according to what this X-ray was showing, the data. Basically, they're trying to fit this model structure to the data. And they tried to go through so many different arrangements. That means it's like playing with the Legos and trying different shapes until they found a shape that fits the X-ray. That's what Watson and Crick did. So, as you can see, the first one here in the mark scheme says, decided to combine what is known the chemical content of DNA with the formation of X-ray. Exactly. So if you just mentioned that it used the X-ray to figure out the DNA content, that's 1 point. I also built scale models, of course, different models of DNA, and they tried to fit them together in a way they agree with the data, as I mentioned, and made several arrangements of the scale model until they found the best one that fit all the data. So this is the points that we get for Watson and Crick. And here are other questions. 2.6 are very short, but if you know the structures of DNA and RNA, then most likely you get easily the questions for 2.6. Now, we have compare, which is basically what are the similarities. We talk about the controversy of Rosenflank, what I just mentioned, or draw just the RNA nucleotides itself. These are future questions that can come up for 2.6. And that's pretty much for 2.6. It's a very short topic. And so these questions that I just put it here are the most seen questions in IB when it comes to 2.6. And that's it.